Dr. O here. In this video, I want to show you the differences between DNA and RNA. Let's go. DNA and RNA are much more alike than they are different, but there are three clear differences that I want to point out here. Quickly, let's just run through them. DNA is double-stranded, whereas RNA is single-stranded. DNA is made of the nucleotides A, C, G, and T, whereas RNA is made of the nucleotides A, C, G, and T's cousin, if you will, U. And DNA has the sugar deoxyribose in its sugar phosphate backbone, whereas RNA has the sugar ribose. Let's dive in a little bit deeper. Here we see a piece of DNA right next to a piece of RNA, so we can do a direct comparison of the single-stranded versus double-stranded nature. So here we see your typical double-stranded DNA. You've got the one sugar phosphate backbone that is bonded to a second sugar phosphate backbone by complementary base pairing between A and T, which we'll get to in a moment, and C and G. Now the bonds that's actually holding these together would be hydrogen bonds. On the right-hand side, we see just the single-stranded RNA. So it still has the blue tape-like sugar phosphate backbone. It still has the nitrogenous bases that make up those nucleotides, but it's not bonded with anything else. Now, let me show you a second image, though. Just because RNA is single-stranded doesn't mean that occasionally we don't see some bonding that occurs. So here we see what happens when you have this single tape-like messenger RNA structure. Occasionally you will have some complementary base pairs bonding with one another to form these temporary double strands, at least small portions of them. So here we see a C pairing with G, an A pairing with U, a U pairing with an A, and a G pairing with a C, all being held together by hydrogen bonds. So just because RNA is single-stranded doesn't mean that you don't see this happen occasionally. So both DNA and RNA are going to form polynucleotides, as you can see over here, which is basically just a fancy way of saying a chain of nucleotides. So you've got your little phosphates here with your sugars. That forms the sugar phosphate backbone. Then you have the nitrogenous bases, which will be either A, T, C, or G, which stands for adenine, thymine, cytosine, and guanine for DNA, or A, C, G, or U for RNA, which would be adenine, cytosine, guanine, or uracil, which is a close relative of thymine. So here you can see all of them. This is all covered in a different video, but your pyrimidines, if you want to remember which three are the pyrimidines, you remember this here. Cut the pi, so pi being PY. So cytosine, uracil, and thymine are going to be your pyrimidines, PY. And then pure as gold is how I remember the purines. Pure, so PU for pure, as gold. A for adenine, G for guanine, but use whatever trick works for you. So those are the differences between the actual nitrogenous bases that make up the nucleotides. The other key difference is here at the bottom, remembering that DNA has the sugar deoxyribose in its sugar phosphate backbone, and RNA has the sugar ribose. So those are the key structural differences between DNA and RNA. Go get your learn on.